At the end of 2019, mysterious warrior pneumonia cases were reported to infect thousands of people in the central city of Wuhan, China. The symptoms of infected patients can vary, but mild cases often experience fever, cough, and fatigue. In some cases, it may cause a severe pneumonia, organ failure, even possible death. In January 2020, the central government of China imposed a lockdown in Wuhan and other cities in Hubei to control the outbreak of COVID-19. A highly contagious respiratory coronavirus known as SARS-CoV-2 is confirmed to cause the pandemic of COVID-19. It can easily infect people through oral fecal route. This leads to a huge burden on healthcare system as hospitals are not able to fulfill the demand of COVID-19 infected individuals. To date, there have been reported 7 million confirmed COVID-induced deaths worldwide. In addition, thousands and millions of people lost their job due to the lockdown. Therefore, economic loss also be one of the negative impact of COVID-19. The advancement of global travel network via land, sea, and air provides a route for SARS-CoV-2 to disperse easily, widely, and rapidly beyond control. The novel coronavirus has evolved for 4 months and rapidly spread to other countries worldwide as a global threat. Vaccination is the best prophylactic method to stop the transmission of infectious SARS-CoV-2 among the community. The currently available COVID-19 vaccines are predominantly mRNA vaccine, adenovirus vector vaccine, and inactivated virus vaccine. However, this vaccine platform requires cold chain maintenance throughout manufacturing and transportation, which increases overall vaccine cost. In addition, COVID-19 vaccines are primarily administered via the intramuscular route. The invasive nature of injectable vaccination may contribute to vaccine hesitancy among some individuals. Moreover, conventional vaccine development often requires a relatively long time frame. Therefore, a cold chain free and fast-track vaccine development platform is an attractive solution that can be used for different infectious pathogens by identifying antigens that induce immune responses. Most COVID-19 vaccines are spike-based vaccines. Yet SARS-CoV-2 has been reported to evolve approximately every 3 months, with major mutation occurring in the spike protein. Therefore, reduced vaccine protective efficacy against emerging SARS-CoV-2 variants has been observed. Consequently, both previously infected patients and the vaccinated population may remain at risk of infection from the new SARS-CoV-2 variant. The idea of solving the repeat evolving SARS-CoV-2 is to develop a bacterial vector DNA vaccine. Recombinant technology plays an important role in this. First, the spike protein of SARS-CoV-2 is the antigen that can elicit the immune responses in recipient. Therefore, the spike protein encoding gene is inserted into an expression plasmid. Then, the recombinant plasmid carrying spike gene is transformed into an attenuated Vibrio cholerae strain, which is known as BIICC2026. And how it works? First, the BIICC2026 P-spike vaccine is drawn by the recipients. The recombinant Vibrio cholerae strain will pass through the esophagus and later enter into the stomach and intestine of the recipient. As the enteropathogen, the recombinant BIICC2026 will colonize in the small intestine and further release either spike gene-inserted plasmid or express spike protein. At last, the foreign antigens are recognized by the recipient immune cells and trigger the immunity against SARS-CoV-2 virus. This vaccine platform offers several advantages over conventional vaccines. First, it supports rapid and highly flexible vaccine development with the capability for quick adaptation to emerging infectious pathogens. Second, the intrinsic self-adjuvant property of the bacterial host can enhance the immunogenicity of the DNA vaccine. Third, the vaccine can be delivered via the mucosal route, which induces efficient mucosal immunity to counteract pathogens at their site of entry. Lastly, its stability without refrigeration makes it a promising cold chain-free vaccine platform. After reviewing the study overview, let's gain further insight from the expert.
Vibrio cholerae vector DNA vaccine platform represents a highly flexible vaccine system that supports the rapid adaptation against emerging infectious pathogens such as Ebola virus, hantavirus, and influenza virus. Its plug-and-play capability allows easy substitution of the target antigen sequence in the shuttle plasmid carried by the BIICC2026 vaccine strain. In this study, the successful expression of the SARS-CoV-2 spike protein was demonstrated in both Vibrio cholerae and mammalian expression systems. This suggests that the platform can facilitate antigen production and delivery by BIICC2026 vaccine strains potentially reducing the expensive purification costs associated with inactivated viral vaccines, naked genetic vaccines, and other conventional vaccine platforms. Moreover, the recombinant vaccine candidate can be delivered via mucosal routes, such as the BIICC2026 bacterial host exhibits good intestinal colonization. Mucosal vaccines are regarded as ideal next-generation vaccines because they can induce effective immune response, IgA, and localized cellular immunity to combat pathogens at their site of entry. Oral delivery may also help to overcome vaccine hesitancy related to needle-based injections. Therefore, Vivo cholerae vector DNA vaccine is a potential mucosal vaccine platform capable of rapid adaptation against pathogens.
Good day, everyone. The recent emergence and spread of highly contagious pathogens such as the hantavirus, monkeypox virus, and the Ebola virus among the population have emphasized the need of the rapid DNA development platform. Okay, the highly flexible of DNA vaccine allows the rapid adaptation and also the fast-track vaccine development. Okay. However, the naked DNA vaccine administration often demonstrated the poor immunogenicity and low delivery efficiency in larger animal models. Okay. Therefore, the BIICC2026 strain is a well-studied Vivo cholerae vaccine candidate, which is safe, non-reactogenic, and well-tolerated in most of the animal models. Okay. And this vaccine can be a very promising DNA delivery vehicles. Okay. And the room temperature stability and the scalable manufacturing of this vaccine strain help to lower the overall cost of the vaccine. In addition, Vivo cholerae also will produce the cholera toxin, which can be served as a potential mucosal adjuvant to enhance the immunogenicity of the DNA vaccine. Thus, the Vivo cholerae vector DNA vaccine platform may serve as a cost-effective vaccine candidate. Okay.
Thanks for the insightful input from the experts. Let us now summarize the key ideas of the study.
In this study, a Vivo cholerae vector COVID-19 DNA vaccine was developed. This platform enables repeat and flexible vaccine development. Its plug-and-play features enable repeat adaptation to emerging infectious pathogen by modifying the antigen encoding sequence in the shuttle plasmid carried by the BIICC2026 vaccine strain. The scalability of production and room temperature stability of the vaccine candidate further reduce vaccine costs, as costly cold chain maintenance is not required during vaccine production, storage, and distribution. This could improve vaccine accessibility in low-income countries, particularly in regions with limited refrigerated infrastructure. However, further evaluation of a suitable formulation buffer is needed to maintain the viability of the vaccine candidates during long-term storage. An optimum dosage and immunization schedule remain to be determined.
Thanks for watching. See you!